So welcome back to part two of my lecture on building molecular devices and machines. I'm going to share my screen. So I'll just grab some slides with you. So we were talking about improvements in the structural validation of artificial DNA-based assemblies, something which I abbreviated in the atomic accuracy challenge. And now I'd like to talk with you about making higher order assemblies. So higher order assemblies, why? I think that's the first question we should address here together. Why would anyone be interested in building higher order assemblies from the bottom up using DNA molecules? Well, first of all, I'm going to give you three arguments. Argument number one is three reasons. Reason number one is integrating components with different functions can create complex machinery. There seems to be like a generic principle. It holds on the macro scale, right? If you think of any, modern day motor, for example, like the one shown here in an exploded view. I mean, it consists of relatively simple components, but they're being put together into a complex structure, which then achieves a motor function. So this is an example of a higher order assembly. But you also have these kind of, this principle also holds on molecular scale actually in nature. So here's an example of a complex molecular machine. It's a depiction of an ATP synthase. And it's actually also a higher order assembly. It consists of multiple protein subunits, which by themselves are relatively comparably powerless, right? They don't really do much. But when you bring them together, well, in this case, they self-assemble into the functional structure, the ATP synthase can actually synthesize ATP molecules. So building higher order objects seems to have, a power, so it's have some power in general. Then the second reason is that we, as a lab, we didn't have specific uses for bigger objects, which we couldn't accomplish with the type of objects we could build when we started with DNA nanotech. And the reason number three is a little bit academic in nature. It's actually not clear what are good strategies and what is the limit in size and complexity when we're building DNA molecules. Maybe there is some kind of limit to the number of components, the overall size, dimensions that you can make with DNA molecules. So it's from that point of view, it's also interesting to pursue the question of making higher order assemblies, ever bigger objects. So what I want to tell you now builds on these two previous works. So in 2015, we completed a project on dynamic DNA assemblies formed by shape complementary non-base pairing 3D components. I will briefly outline what we did there. And then we built on this and another then worked on the Giga-Dalton scale shape programmable DNA assemblies. So I'll only touch on this. So I think from my point of view, I'm subjective. These are very interesting papers. I invite you to take a closer look into them. Okay. So let us talk about the Hubble and the hole. So the Hubble is actually a German dialect for a protrusion. So and one of my students, Klaus Ragbauer, is the guy in the center here. He is from Niederbayern, which is like a lower Bavaria, and they had this peculiar dialect, and he used to call everything which was a protrusion would call the Hubble. So and this, what I want to tell you now, is a sort of a new concept where we literally copied lock and key shape complementary interaction mechanisms from the protein world to the world of DNA nanotech. So because in fact, if you look into nature, you'll find that actually I need to rewind a little bit, right? We're familiar with shape complementarity as a way of docking protein subunits together. But when we think of nucleic acids, we mostly think of base pairing, right? Sort of as a way of bringing together multiple DNA strands. So, but as a matter of fact, actually, there are also nucleic acid structures known from nature, which do not interact via base pairing, but via some kind of docking, as we know from proteins. So here's an example of such a system. So this is RNase-P, which is a RNA-based enzyme. Um, and what you see here is a mature tRNA, a transfer RNA, and it has this party of the molecule which is terminated with two base pairs. And then it turns out that this tRNA actually can dock into this pocket in the RNase-P, and it's held there based on stacking interactions. There's a precise fit, it just fits in there, and sort of the shape is such that it's perfectly complementary, and then it can be held there by stacking interaction. And then RNase-P processes the tRNA, and I think it cleaves off this overhang here, and then the tRNA can do its job. So then basically what we were inspired by this, we invented this system here, which basically, so the gist of it is sort of you make DNA nanostructures which have topographic features, sort of as realized by a certain pattern of blunt-ended DNA double helices which are fixed onto a surface. And then if you want to, we design a binding partner such that then you have basically a little recess which precisely corresponds to this blunt-ended DNA protrusion on another component. And then the idea here is that these things can actually stick together simply by fitting these, by docking these protrusions into these recesses, and then rather than base pairing at the edges of these blunt-ended DNA, you can get stacking interactions, and which are sufficiently strong to actually hold to stabilize a bound complex. This actually works. So you can look at the data and see in this paper, we tested this on a variety of objects, and you can do multimerizations, shape-specific multimerizations. You can do, make switchable objects. We'll talk about this later a little bit more in detail, where the different conformation states are stabilized by this kind of shape complementary stacking interactions. So the cool thing about the system is that you no longer need single strands in the system. You can basically make an object, incorporate all the DNA single strands into a fully folded object. It doesn't have any free DNA single strands, and then merely by simple arguments of geometry, can program a structure to specifically interact with another. You can program an object A to specifically interact with another object B. The only argument you need there is basically a geometry one. How can you plug this object into this one and make sure that no other object can actually also fit in the same binding pocket. And something that maybe not everybody appreciates is that DNA single strands are always somehow even weakly inherently sticky for other DNA single strands. So even if DNA single strands are not complementary to each other, they kind of want to stick to each other if you have enough of them. So going to larger and larger objects, you want to have a system which doesn't have DNA single strands anymore, and this is a path to get there. So and then sort of with this base pairing or base pair stacking approach, we went into building multi machines, something I'll talk about later. Stuff that can be switched for different conformational states. And we tried to use this method to build ever larger objects. And it turned out to be a very powerful approach. So the first thing that we did is then we asked ourselves, can we build finite size higher order objects based on geometrical arguments? So here's an example of such an object that we built. Is basically, consists of several subunits, like, DNA origami based bricks, which have a certain pattern of recesses and protrusions, which are such that sort of the differently colored objects interact with each other in a specific way. You can literally stick them together, and they're held together by stacking interactions. So, and then with this design approach, we were able to build massive objects on the bottom up, which approach the Giga-Dalton multi-mass range. So, This is an example. 3D structure, as we planned it, consists of many of such objects. And here's an experimental realization. So this object actually did assemble successfully, and its whole point was to make something well-defined but massive. And this particular object, which sort of, you see an image here, is recorded in negative staining by negative staining transmission electron microscopy. It actually contains a total number of 220 viral genomes and 50,000 oligonucleotides. A total of 1.8 million base pairs, and as such, it has a molecular mass of 1.3 gigadaltons. So it's a massive molecular object, and the entire shape still encoded by sequences. So by using a hierarchical approach, first you program sequences to form well-defined brick subunits, and these bricks interact with each other in a particular way to make this higher order object. If you want to know more of the details, I invite you to look at the papers. I just wanted to get this out of the way so that I can now explain a more detail, a higher order assembly, which we made recently, which is not yet published, that actually has a purpose. So what I want to talk about is this project. It's called Programmable Icosahedral Shell System for Virus Trapping, also known as the Venus Virus Trap. It's one of the biggest collaborative projects which we have pursued so far. The authors you called David and White are from my lab. Then we have collaborated here with virologists from the Hammels Center in Munich, the German Center for Infection Research. And then the authors called it in purple, biophysicists, soft matter experts from self-assembly experts from Brandeis University, which helped us understanding viral self-assembly principles. So and these are sort of the lead people in my lab who contributed to this work. It's Christian Siegel, PhD student, and Anna Wilner, PhD student, Dr. Walter Engel, and Dr. Jessica Kretzmann are two postdocs. So let me first outline the purpose of this whole research project. So the vast majority of viral diseases actually have no cure. We all experience this now at the moment in the pandemic, the COVID-19 pandemic, and instead, viral diseases are typically being addressed in the term by prevention, basically, right? So people ideally at some point will have a vaccine which can make us immune against a particular viral disease. But developing vaccines takes time, and it would also be actually really nice if we had a strategy to make a cure, an actual therapy, if you had an acute viral infection of a certain type. I think over 80% of World Health Organization listed viral diseases really have no cure. There's no, you cannot take any drug to help you if you had that viral disease and you don't have prior immunity acquired by some vaccination. So this is a real problem. So we had an idea here for a potential generic antiviral platform. So what if you could build a structure that you could design such that it could specifically engulf a viral pathogen and thereby deactivate it, thereby basically preventing it from interacting with host cells, and you would basically support the body and giving it more time to develop an adaptive immune response to get rid of the viral pathogen, simply by, if you trap enough pathogens in such cages so that you take them out of the system, you could reduce the viral load and thereby contribute to thereby achieve a therapeutic effect. So that's the underlying concept. In a way, it has similarities to the function of neutralizing antibodies, because they bind to viral pathogens in a way that the viral pathogen can no longer, or is inhibited from, at some step in the life cycle, For example, it's inhibited from interacting with host cells. So that's a function that this virus trap, could accomplish. So, and actually we have a prototype of a working virus trap, which I'll really show you so that you maybe to spike your curiosity about this. So what you see here is a fluorescence microscopy image of a mammalian cell culture. So all the green objects actually are a human cell, cultivated human cells, and they are green because they express green fluorescent protein. And they do this because they have been infected with a model virus, in this case, adeno-associated virus, which was um, um, which was modified so that upon infection, affected cell would sort of express green fluorescent protein. So all of these cells, the cells culture had been incubated with a certain dose of these adeno-associated viruses, and all of the cells practically got infected. Now, we made a virus trap which could specifically trap this adeno-associated viruses. And if we apply the same dose of these viruses with our virus trap to the cell culture, basically much fewer cells now turn green, which means we actually do have a neutralizing functions of these virus traps, at least in cell culture. That's very promising. So now I'd like to tell you about the virus trap. So the virus trap, basically, it's a massive market engineering challenge, because in order to build such a shell, we want to build a shell which is as large and even larger than an actual viral pathogen, and we want to build this shell around the virus. So here's how we solve this problem. So we designed triangular DNA-based building blocks and solved cryo-EM structures of them. So here's one example of such a cryo-EM structure, such a triangular subunit. The edges are beveled with a particular bevel angle, so that when multiple copies of these triangles come together, they will develop a curvature, and the curvature is such that it will precisely give a closed shell with a sort of number of triangular subunits, which is sort of defined by the bevel angle. And then if you look at these edges, you can see there's a recess, and there is also a protrusion here. And here all the edges are equal. So both copies of these triangles can actually interact with each other. And when these recesses fit in the protrusions, stacking interactions at the interfaces can engage and can hold stabilized sort of higher order assemblies. So this is an equilateral triangle design. It will form an icosahedron, as you will see, with 20 copies of these triangles. And this is an isosceles triangle design, also a cryo-EM structure, so it's actual data, right? This edge is longer here, the bottom edge. And also the sides look, the edges here look different. This only is a protrusion. This edge only features a recess. So there's a rule that says in which way copies of this triangle should multimerize. So this edge will always interact with this edge, whereas this one actually will interact with itself with a rotated copy of this triangle. Okay, so basically what we have triangular subunits, and by simple geometry, which is encoded in the design of these triangles, namely bevel angle and interaction pattern, we can make these triangles self-assemble into higher order shells, like those shown here. Again, what you see here is data. So these are cryo-EM electron density maps from actual experiments. So these aren't schemes or anything like this. So then we have your different design variants. Here is this icosahedral triangle that I meant. It makes icosahedra. This is this isosceles triangle. It makes a bigger shell with 60 copies of this triangle in it. And this is an even bigger shell, which actually takes notability. You need two different types of triangles. So these are the kind of shells you can make. You can actually modify these shells further, just for completeness. You could add additional modules onto these icosahedral shells if you wanted to, and to, for example, to plug the holes here. So you could, this is kind of a little bit holy. So if you want to get rid of the central cavity, you could add additional module. And this again is a cryo-EM structure. This object actually exists. So and then just to give you an impression how well this works, this is a raw cryo-EM micrograph with the T equals 3 shells. in it. So you see several of these particles. They look very nice and roundish. And there's a little bit of debris here, some triangles, some partial fragments. But overall, um, I mean, the yield is really nice. When we design these triangles in the proper way, they assemble the desired shell and nothing else, or very little nothing else. And to get there, we had to spend quite a little time in fixing these designs. So the assembly success really depends critically on the monomer quality, and iterative design approaches help validating and refining the subunit design. Um, so just give you one example. So we had like one version of these equilateral triangles, which used to have sort of a systematic folding defect in one of its vertices, and because of this folding defect, it wouldn't multimerize and give higher order objects. And then you can scratch your head, why is that not happening? Well, only because we had the structure, we could discover that something was wrong in the folding. We looked into the design, there's some details here, and we could fix it, and then made a new version of this, which now all the edges were perfect, and then it readily multimerized in the context of these shells. Also, the geometry matters quite a bit. So we did angle variations of these bevel angles, and only if you hit the right angle, it will make the target shell. But if you deviate from this target angle by plus or minus 5 degrees, then you get a mixture of different products. So, for example, for the design that's supposed to make triangles, if you pick the wrong bevel angle, like you make it a little bit too small because the curvature isn't strong enough, then it will make it cannot actually form the icosahedra because it doesn't get close. It requires deformation, and if you err on the other side, it will collapse into making octahedra, so too small object. Okay, so then let's have a closer look onto these shells. So this is a cut through electron density map acquired for this icosahedral shell, which consists of 20 origami triangles. There's a thick, robust wall. You see the circular cross section. These are the cross sections of individual DNA double helices. And the internal cavity is about 80 nanometers. So here there's quite a number of natural viruses which could actually, nicely fit into this cavity. Then here's the bigger shell, the T equals 3 shell. So the EM density map doesn't have the same resolution like the vertical resolution, so we can no longer see the individual helices. But still, it's a big, nice cavity. It's 150 nanometer diameter, and actually, you can fit this icosahedron. You can fit the icosahedron into this bigger shell, and the icosahedron actually roughly corresponds to the diameter of a coronavirus, for example. So the T equals 3 shell could be a candidate system for trapping maybe a coronavirus inside, which leads us to the next question, which is, how do we actually trap virus particles in the shells? So, I mean, I talked about making shells, but if the shells are closed, you can't get anything inside. So, there's two ways you could approach this problem. One is to build virus trapping shells that have sufficiently large apertures for which virus particles could diffuse in and then, get stuck there with additional functionalities, or you could dynamically assemble the shells around the viruses. So we worked on both aspects. We made most progress with this strategy, so this is what I will talk about today. So how do we make such gigantic shells with apertures? So the icosahedron I talked about is a fully symmetric object, right? So you cannot individually address a particular triangle, they're all the same. In 1962, Kasper and Klug outlined the symmetry principles that underlie most spherical viruses, and they did this with the help of hexagonal net projection. So where each of these triangles represent one of the icosahedral faces in the viral capsid. So, and this is the inspiration for our design concept actually in the first place. So for the icosahedron, for example, we would design a triangle which can interact with all its edges, right? And so then basically what happens during assembly is that all of these positions on this hexagonal net, like in the thought experiment, is being populated by triangles. And then, they have all these interactions, by the edges, so this thing will fold up and form a complete icosahedron. So now if we want to make an icosahedron with an aperture, for example, what we could do is we take one set of triangles and sort of modify it such that it can still undergo these edge-to-edge interactions, but maybe you can shut off the interactions and thereby inhibit further growth in this direction on the icosahedral net. Then you would get a pentagonal vertex of an icosahedron if you would do that. You could add in a second type of triangle, which you would program to interact only with this particular edge of the blue triangles, and then you would get sort of a half icosahedron. You can add a third type of triangle, which would be designed to only interact with these edges of the orange type of triangles. Then you could form a sort of, like a icosahedron which misses one pentagonal vertex dome. So it looks like maybe like a bowl with a somewhat smaller aperture on the top. So, and this actually works. So what you see here is an icosahedron, is a cryo-EM reconstruction of the half icosahedron. So it's basically two copies of this icosahedron would make one icosahedron. I think it's a pretty cool structure actually. It's 10 DNA origami in a sort of finite size higher object, and it just forms by geometry arguments. And then here there's a cryo-EM map of the sort of 15 triangle containing incomplete icosahedron, which misses one pentagonal vertex. It's a bit floppy on the side. That's why this sort of, it looks a bit messed up here. It's just that there's the density. There's not enough density in the structure. But yeah, overall this works. So, and now sort of we have a structural basis to trap viruses. So how do we get them in there in the first place? How do we make these shells sticky for a target virus, specifically sticky for a target virus? So, well, there are multiple strategies you could consider. We used mostly antibodies at this point. So there's macromolecules which have specific affinity for viral surface proteins, many of them available. What you can do is sort of you can tag them with a DNA strand, and then use this DNA tagged antibody and hybridize it to single-strand DNA protrusions on the interior surface of these cavities, of these shells. But there's other options. You can think about, using host receptor domains, put them there, basically, make like a decoy target. If for example, the virus A attacks ACE2, ACE2 domains on host cells, you could make the ACE2 domains, as it's now for COV2, for example, and conjugate them to the inner surface of these shells. You could also use peptides. There's peptides that are known to be bound by certain viruses. So there's aptamers. So you could actually generate aptamers without knowing much about the structural details of the target virus, and that will be sort of the easiest target, as the easiest candidate for coating inner surface to make it specifically sticky for a target virus. But if you use antibodies for our proof of concept study here. So what you see here is a negative staining TM micrograph of a half octahedra built from DNA shells, which have been functionalized with an anti-hepatitis B virus antibody, and then our octahedral, half octahedra actually do trap hepatitis B virus core capsids. This looks very nice, and you can see this on the side. These are the virus capsids, and these are the triangles here. And you can use a bigger shell, for example, this T1, this 15 triangle containing icosahedral shell is big enough that it can accommodate up to 3 of these hepatitis B core capsids, so you can soak it up basically. And this why we sort of call this the Venus virus trap. It's like fly paper, so the viruses, once they're in, they cannot get out. This is a cryo map of a hepatitis B core capsid in the center, which has been sequestered and is coordinated by 2 half octahedra, basically fully enclosed. And I think you would agree, hopefully, with me that it's hard to see how such a trapped virus could still interact with host cells. So we think that in this configuration, maybe you can make a different one, which sort of even closer packed. I mean, it will be hard for the virus particle to integrate with the cell. And here, how is it held there in the middle? Well, it's a little bit hard to see in the EM map, but there's the density in between the virus and the DNA walls, and this is the antibodies which are being used to trap the virus. So just trapping these viruses in these shells actually prevent the viruses from undergoing virus surface interactions. So we did various types of experiments to study this. So the first one is entirely in vitro. We have here an anti-hepatitis B virus core capsid antibody, which we coated the surface, and then we'll do. You can think of this as a type of ELISA experiment. We wash a virus sample over the surface, and then if these viruses, they can get trapped by the antibodies, and then you do a second wash where you apply another antibody for the virus, which is conjugated to a horseradish peroxidase. So then horseradish peroxidase can convert initially colorless substrate into a colored substrate. So you get sort of, depending on how much viruses have been bound to the surface, you get more or less colored product per unit time. That's how you can quantify how many viruses you actually have stuck on the surface. So then what we did is we compared naked viruses with viruses that had been trapped in a variety of conditions with such engulfing shells. And the question is, to which extent does adding the shells actually suppress the ability of the viruses to stick to the surface? And also, to which extent is that actually better than using just antibodies alone? It's an important question too. So here's the result of this experiment. It's busy graphs, so I'd like to take you through this. So first of all, we're going to see on the y-axis, this is the extent of virus blocking caused by the shells. And this is plotted over the number of DNA shells that we have added per hepatitis B viral capsid. Yes. And the number of antibodies which we use per half shell is also given here. So let's look at all data points. So here, the first data point here is where we just had antibodies and the hepatitis B virus capsids in solution. So, and in these particular conditions, we have about 1 nanomolar of the hepatitis B binding antibody, and we have 2.5 picomolar of the hepatitis B core capsid, which means that the antibody is in a 400 to 1-fold excess over the hepatitis B core capsids. Still, even though the antibody is in such big excess over the capsid, you see there's very little blockade going on. So the antibodies, even though they're known to specifically stick to the capsid, because they can individually bind and unbind, they don't completely and efficiently cover the surface to such extent that they can actually, substantially prevent the virus from interacting with the surface. But that's actually something that you would want to have. You want to have an agent which really prevents the virus from undergoing interactions with the surface. And the antibodies, even though they already have them at a super large excess over the viruses, they don't accomplish this function. And now, if we take the virus again, and now our non-specific shells, just shells which have not been functionalized with antibodies, they also don't do anything. Why would they? Well, there is no functionality in them that would specifically trap the viruses, okay? So now if we bring these two components together, shells plus antibodies, and we basically, we see that if we put the antibodies in the shells, the more shells we put into the system, the more efficient is the blockade. So here we are actually at the ratio of about 2 shells per hepatitis B capsid in the system, and then we get about 100% shows 100% of virus blocking, which really shows us that, you know, uh, first of all, we have the antibodies in the shells. They promote the sticking to the capsids, but the shells really, contribute to keep preventing the virus from interacting with the surface, full occlusion presumably. And then if you look at these other data points here, at this data point, for example, we only have approximately 4 antibodies per shell in this circumstance, and we still have 80% of virus blocking. So this shows us that sort of for multivalent binding, we get still stable binding, and the material of the shell really contributes to the passivation of the virus. So this is very promising data, and it's sort of biophysical analysis. That's why I sort of spent some time to explain it to you. So it tells us virus occlusion works in vitro, and that's why we moved on to test this with actual live cells. So we did the following assay. I really touched on this in the introduction of this part. So we took a human cell line, and in its pristine form, it's non-fluorescent. So then we will add adeno-associated viruses carrying an EGFB gene. These are infectious but non-replicative model viruses. If they infect the cells, cells will express EGFB, so they turn green. So then the experiment we did is we compare to which extent trapping in the viruses that carry the EGFB gene, to which extent it can actually suppress the expression of GFP in these cell cultures. So, and here is some microscopy data showing this. So if you have the viruses and no neutralizing agent in solution at all, almost our conditions that we picked here, we get almost 80%. Almost all of the cells basically are being are turning green. So these are all the nuclei, and then I think close to 90% of all the cells are actually turning green under these conditions. Now, if we add our shells to an experiment where we have added the exact same amount of viruses, shells that are made specifically sticky for adeno-associated viruses, again using antibody, then very little cells remain that actually turn green. Here are the nuclei, the fluorescent channel. Very few of these cells are actually green. So we did successfully neutralize the virus in this experiment. And just a comparison, you can ask the question, well, maybe it was just the antibodies that neutralized the viruses, not the shells. If you just use antibodies, the free antibodies, at the same concentration, which were effectively where they were used here in this experiment, you have many more cells that still are turning green. And this is because, again, you get this effect that individual antibodies, they may have nice affinity, but they can still individually unbind and rebind to the virus. And therefore, there's still room for the virus to actually get into the cell. This is shut off in this, or improved in the experiment where we actually add our shells. So yes, the half shells of the antibodies functionalize for adeno-associated virus. They actually can neutralize these AAVs in cell culture. It's nice. Another question you could have is maybe we kill the cells with the shells, and that doesn't seem to be the case with cell viability assay. And at all of the concentrations at which we tested our shells, they didn't have an effect on the cell viability, so that's good. So I'm going to venture and say, based on the data we have, our virus trap concept works with live cells, and now we're going to move on and need to see whether this actually works in actual organisms. But I'd like to still talk about this one point. So I got this question a couple of times in the past. Does this concept actually have any therapeutic merit in the first place? Well, blocking virus services can have therapeutic effects. That's I think fairly well established. So there's multiple points that show this. So there's, for example, it has been seen that critically ill patients that were sick with COVID-19, they actually improve once they've been treated with plasma taken from convalescent patients, or patients which had COVID-19 but recovered. So presumably this improvement is due to the presence of neutralizing antibodies, which reduce the viral load then in the acute infection. So then monoclonal antibody therapy is a strategy that's actively pursued as a potential therapeutic intervention for COVID-19. I think President Trump received such treatment when he was sick of COVID-19, and at least he claimed that this was miraculous. And then there's basically a whole bunch of strategies which basically is aimed toward blocking viral surfaces. So there's a nice paper from the Baker Lab where they designed mini protein inhibitors which target surface proteins individually with the aim of passivating them and preventing these surface proteins from interacting with host cells. So we're trying to do the same, but we have really this brute force approach, making a massive microobject which traps the entire virus, and then we're shutting off entirely all the ways for interacting with cells. And then you could ask, well, why not just use antibodies? Well, I hope I could sort of, I tried to touch on this in my slides a little bit. So, well, we've seen sort of in our assays that sort of individual antibodies, because they can individually unbind and rebind, you need a lot of antibodies to really cover the surface. And, first of all, you need a specific type of antibody for a therapeutic effect. You need an antibody which needs to be neutralizing. It needs to bind on the viral antigen in such a geometry at such a spot where it can actually interfere with the viral surface protein binding with the host cell. And developing such neutralizing antibodies takes time. And we're not saying we're going to replace antibodies. We're saying we could improve maybe the efficacy of existing antibodies. Maybe you have some antibody for a target viral pathogen, but it's not neutralizing. Then when you put this into a shell, you can make it into a neutralizing system. And then we have also avidity, multivalent binding, plus occlusion with these massive shells. And then as a third point, is an interesting aspect, there's something, an effect that's called antibody-dependent infection enhancement. Maybe this can be avoided by including the Fc fragments of antibodies of our shells. Okay, so next steps for us with this venous virus trap is to test this in actual organisms. We could maybe make some omnivore virus traps, which meaning like shells, which are functionalized so that they can target multiple viruses in parallel. And of course, we can include additional functionalities. You could think about putting in proteinases or phospholipases, basically enzymes which degrade virus proteins, and thereby basically really actively contributing to getting rid of this viral pathogen in the body. So I'm really excited about this direction. I hope you're excited about it too, but of course there's a lot of technical risk associated with this yet, and we need to do the groundwork and test this very thoroughly to see where it actually can have a real effect in the real world. So there's a consortium, European Commission funding consortium, which I'm coordinating, including virologists and physicians and chemists and us. And we're trying to make is a real therapeutic strategy. Okay, I'll take a short break here.